Alice Valentini. I am a research engineer at Spygen, and I'm also Spygen co-founder. And I will talk about eDNA for aquatic biodiversity monitoring. So just a small slide to present Spygen. So we created in 2011. And we are now a mission-led company, and we have as a goal to improve biodiversity monitoring and conservation using environmental DNA. So we provide analysis of DNA in the lab. We also have some sampling tools, like the little drone over there in the middle of the slide, and also sampling kits similar to those that Kate showed. But our kit allows to sampling a large volume of water up to 30 liters. So I don't give an introduction about environmental DNA because Kate already did very well. So I will go directly on some example how eDNA can be used. So there is two techniques for environmental DNA analysis. One is the eDNA barcoding approach. So you go to the field, you sample your environmental sample, in this case of water, you do the extraction. In this case, you do the amplification with specific primer for a target species, and you can see the results or on a qPCR or just on a gel. And this technique, we use this technique for the detection of great crested newt in UK. Great crested newt is a species that is particularly interesting in UK. So we tested how eDNA can enhance its detection. So we compare our methods using traditional methods in 35 ponds that were visited 4 times. And we showed that environmental DNA was higher detection probability than all the other traditional methods used at the times. And so we go further, and we decide to give us our sampling kit to volunteers. They were not trained or supervised, but they only have the kit and a written protocol, and they go around the UK to sample all the ponds where a great crested newt was known in an area of 1,000 kilometers around. So at the end, we were able to find in 219 sites, more or less more than 90%, we correctly detected the great crested newt, meaning that these techniques can be easy to implement in the field, even for not trained people, and also like volunteer, like Kate explained before. So the second part will be more about eDNA meta-barcoding, and Kate already explained how this works. So this is the same as the eDNA barcoding, but at this time we use group-specific primers for a group of species like fish or amphibians. We do the sequencing using next generation, and we compare the output of the sequencing with a genetic reference database, and we have a list of taxa. We use these techniques to describe the fish biodiversity through the Rhone River. So actually we sampled the Rhone River from the Swiss border through the Mediterranean Sea. So we use our sampling engine was a drone that allowed to sample for 30 minutes on the water, that can be trained by a boat or just slowed down from a bridge. And so we collected all around, so we have more or less 200 samples, and we compare the eDNA results with what was obtained with electrofishing each year. So it's possible to see in the blue dots represents the mean species richness detected with electrofishing per each year. The triangle represents the fish species richness on 10 years cumulated, and the red squares represent what we obtain with only 2 eDNA samples in 1 year. So we arrived to, only 2 eDNA samples. We have the exact species freshness that was achieved in 10 years of electrofishing in this big river. And we will also be able to detect 2 species that were critically endangered. One is a zingle asper, is an endemic fish species in the Rhone Basin. It was thought to be extinguished in the river, and also Misgurnus fossilis. So I show 2 examples of what's obtained in the temperate region, and I will show other examples on how eDNA can be applied to tropical samples. So we start to work on fish in French Guiana since 2014. And actually the problematic here is that it's not possible to do traditional survey, because in a small stream especially, the traditional survey use retinone that kill all the fish present in the river, and so it's not possible to use this technique anymore. So eDNA be thought to be an alternative, non-invasive. So we sample more or less all the river drainage, the Guyana territory, and for 2 rivers, the Maroni River and the Oyapock River, we do exactly the same experiment that we did for the Rhone. So we sample regularly, more or less every 10 kilometers, all along from the upstreams through the estuary in the Atlantic. And as Kate said in his presentation, when you have an eDNA samples, you can do fish, but you can take the samples and do a second analysis later on. So we were interested to see which mammal were present in the Maroni River. So this is an example of few species that we found more species, but this is an example of the most interesting one. So we were able to spot giant otter, jaguar, white-lipped peccary, and mantine. So for French Guiana, we analyze the fish. We also duplicate this analysis in Casamance River in Senegal. And at that time, we were interested on the study all vertebrate species. So we only want 2 samples of eDNA. We were able to find all the vertebrate species present in the different part of the Casamance River, with more marine species present close to the estuary and more terrestrial species present on the inner part of the river. So we are able to distinguish shark, ray, mammals, fish, reptiles, and birds. We did also, we were interested to see how eDNA metabarcoding was able to describe high diverse tropical forests biodiversity. So we collaborate with TAPESP in Brazil to detect amphibian species in Pechinguawa Natural Park, next to Sao Paulo. So these are the typical stream present in the region. So we were able to describe the amphibian population that were living in those rivers, or that just pass by, or they use it only for the reproduction. And the eDNA was much more have a much higher detection probability of the species, because in parallel, the traditional sampling method find only 3 species, and we were able to find all the 9 species that were present in the region, even those that were considered very, very rare. So environmental DNA is particularly useful to find rare and elusive species. So I will show you 2 examples of these. One is the one that we did in the Mekong River, that we sampled 6 deep pools in the river from Laos to Cambodia. And we were able to spot out the critically endangered Mekong giant catfish in one of the pools. And in another deep pool, we were able to find the iridium dolphin. And this report was quite surprising because fishermen in this region said that they found they looked on the water, they saw giant catfish, but actually probably they misidentified the fish with the dolphin. And also, it's possible to find very, very elusive species like the giant muntjac. In this study, we took water sample from this kind of river or on the small forest pond, and we analyzed the DNA of mammals, and it was possible to spot several different mammals that come to drink in those water, and also the giant muntjac. So just to conclude quickly, eDNA is a powerful tool for species detection, can optimize the detection and to monitor rare, endangered, and threatened species because it's very precise and very powerful. Also early detection of alien species, and eDNA inventory can serve for reliable monitoring for ecosystem health, ecosystem functioning, and servicing, and provide strong indicator for early detection of species decline on biodiversity erosion. And thank you very much.
Wonderful, thanks so much, Alice. It's fascinating. There are any, I know there's been chat going on around, actually I wonder, Marco, I know Kat offered an answer, but do you want to jump in and ask your questions from before? I'll take that as a no. Okay. Oh. Karen, do you have a microphone?
Yes, I do.
Do you want to jump in?
Yeah, I was just interested. I've done some work with Patricia Medici, a taper specialist group, and she traps lowland tapers and takes all sorts of samples for DNA analysis and parasites and all sorts of things. But I was wondering, is it possible to match the results of DNA testing for individuals to samples from rivers to identify an individual?
From rivers, not, but there was a study on shark whales that they demonstrated that it was possible to distinguish different haplotypes and to describe the population. And I think Arnaud will talk a little bit later on in his presentation about this.
Oh, wonderful, thank you.